Well, welcome back, everybody. And it is a great honor to present Dr. Christof Koch. Dr. Christof Koch is a German-American neuroscientist, best known for his studies and writings exploring the basis of consciousness. And he was trained as a physicist and spent 27 years as a professor of biology and engineering at the California Institute of Technology in Pasadena. And he is now a chief scientist of the Mind program at the Allen Institute in Seattle, leading a 10-year large-scale, high-throughput effort to build brain observatories to map, analyze, and understand the mouse and human cerebral cortex. On a quest to understand the physical roots of consciousness, Dr. Christof Koch published his first paper on the neural correlates of consciousness with the molecular biologist Francis Crick 30 years ago. Dr. Koch is interested in mystical experiences and what they can tell us about the mind and the brain. He's a vegetarian and cyclist who lives in Seattle, loves big dogs, biking, climbing, and drawing. He is an author of a few popular books, and the latest one is called The Feeling of Life Itself, Why Consciousness is Widespread but Can't Be Computed. For more information, please visit his website, www.christofkoch.com. And today, Christof will tell us about the neural correlates of consciousness. Welcome, Christof.
Thank you. Thank you, Angelina. So I'll talk to you about two subjects. I'll talk to you about the neuronal correlates of consciousness, experimental approaches that we can take using today's technology to explore the footprints of consciousness in the brain, the human brain or animal brains. And then I'll talk to you about an ongoing adversarial collaboration to test predictions between the two dominant or two very popular scientific theories of consciousness, integrated information theory that Pervo just mentioned a couple of times, and global neuronal workspace, and some of the implications of those theories. But there again, I'll focus on experimentally accessible predictions. Could I have the first slide, please, Angelina? Yeah, if you can go into whole slide mode. All right, so first slide. Next one. Yeah, if you can just click through those four. So for me, consciousness is very simple to define as any experience. So it could be an experience of Rodin's thinker looking at something, like right now I'm looking at my screen here, seeing my slides, or you could reflect back on what I just saw, or you could imagine something. You could think about thinking, right? You can become self-conscious, particularly human adults can do that, particularly if they're literary, and then they can write books about it. Or there's this interesting class of experiences when you seem to be highly conscious, but you're not conscious of anything in particular, or the content of your current state of consciousness at that point is very minimal. In the limit, there may be no content, what some people refer to as a pure experience. You can have these during religious conversion, during near-death experiences, in flotation tanks. These have been frequently reported when you take certain drugs of the serotonin class, so-called entheogens like psilocybin, DMT, or 5-MeO DMT, the toad. You can get these very intense states of pure experience. But what's common to all of them, they all share a commonality. They all feel like something, in the famous word of the American philosopher Thomas Nagel. I don't think really these are higher order of consciousness. These are just different conscious states, some more common that we experience every day, some less common. Next slide. What Francis Crick, when we were interested in consciousness and wrote more than a dozen of papers in the 1990s and early 2000s, was interested in is moving the debate from either philosophy or from exotic physics. In other words, physics that's not easy to test in biological systems, particularly in living systems that live at that are hot by physics standards at 300 degrees Kelvin in a highly aqueous and wet environment, to move those debates into something more experimentally accessible, where we can actually make certain predictions. And so this was this idea that so-called the neural correlates of consciousness, or also abbreviated as NCC. So the idea is for every conscious experience, so those conscious experiences I just talked about, there will be an NCC. We know it's in the brain, it's not in the heart, as most cultures used to think. It's in the brain, and for every conscious percept, for every conscious experience, there will be such an NCC. And we can ask what's common among them. So what's common among the NCC for seeing a dog versus seeing a cat, or seeing red versus seeing blue? Are they in the same part of the brain? Do they involve similar neurons? Or when I hear something versus see something versus smell something versus become angry, is the NCC in all of this, is it always similar type of neurons? Do they have some commonality in terms of the architecture, in terms of where they are in the brain, in terms of the type of neurons activated, the genes involved, the neurotransmitter involved, the type of neurons involved, layer 5 versus layer 6, global workspace neurons. So these are all different ideas regarding the NCC. Furthermore, it's a causal notion. It's not just a correlation. The idea is also it should have causal properties. In particular, if I artificially induce the NCC, for instance, by using a surgeon's microelectrode, by transcranial magnetic stimulation, by drugs, by optogenetics, etc., I will induce the conscious percept. And if I inactivate the NCC again by some artificial means, you will eliminate the percept, even though, for example, the stimulus may still be present. And today, of course, we have some very powerful tools in neuroscience to manipulate NCCs. Next slide. And so one way to study the NCCs, there's sort of two broad classes of experiments. One is this here, where you identify the content-specific NCC. So here it's vision. People have also done it with somatosensory or with auditory stimulus, but here I sort of schematically demonstrate a visual NCC experiment. So I flash on very briefly that picture, that chiaroscuro picture you can barely see of a woman's face. It's flashed up very briefly, let's say 20 milliseconds. It is so brief that only half the time you will see it, for a variety of reasons, not under experimental control, in fact, not even under your own voluntary control. When I deal with very briefly presented stimuli, sometimes you see them and sometimes not. And so I can arrange it experimentally by manipulating time, duration, contrast, etc., that you see, half the time you see it, you actually have a conscious experience of a face, and half the time you do not see a face. You still see something. So here the contrast is between seeing a face versus seeing just this noisy pattern. Okay, so it's not, here the contrast is not seeing versus not seeing at all. It's seeing one thing, conscious face versus seeing something else. And I can, collect all the trials on which you saw something, and I can compare that to your brain responses when you didn't see it. Here it's done for fMRI, but you can do it using EEG or microelectrode or MEG, whatever. And here you can see, for instance, so there's a brain, if you do the contrast, you see brain regions outside primary visual cortex that are active. You see neurons or brain activity in the fusiform phase gyrus, and you see activity in premotor areas, prefrontal areas, okay? But now you have to worry, well, this is not only involves consciousness, for instance, it also involves a button press. Maybe I'm picking up the neuronal response of the button press. It also involves the fact that I have to do a task and I have to remember the task, right? So I'm sitting in the magnet carrying out a task, which is a different state than when you're just, not doing anything and suddenly you become conscious of something. So experimentalists, and this is the art and skill of doing experiments, have to account for all of that. Next slide. And so here, for example, they have done this using so-called no report paradigm. So here you control for the task and you control for the button presses by having spontaneous reversion. In this case, it's a slightly different experiment. Here you have an experiment that consists out of two gratings that move in opposite direction, left or right. One is green and one is red. And sometimes, because of the property known as binocular rivalry, the two stimulus sort of fight for dominance in your brain, as it were, and at any given point of time, you only see one or the other, and this happens spontaneous. And you can track this not only by people telling you, I saw the green moving to the left or the red moving to the right, but there's actually a thing called optokinetic nystagmus, an automatic response of your eye oculomotor system that will always track the dominant percept. And so even without an explicit task, you can still see what people, whether the subject currently sees the left or the rightward stimulus. So here, this is one way people have invented how you can control for some of these variables. Next slide. And then, so this is the no report paradigm. And then if you do this in this case, you see that the activity in the front of the brain seems to go away. So the inference was the activity in this case in the front of the brain has to do with the task setup, but not with pure consciousness by itself. And so this is the experimental art. This constantly refines, becomes better to really focus on what you want. So for instance, you also have to distinguish selective attention from particular selective visual attention from visual consciousness, because those are not the same thing. And we now know that through roughly 50 different experiments that you can attend to things, you can pay attention, you selectively process a certain aspect of the visual stimulus without becoming conscious of it. So this is an important thing, to really focus on the conscious experience and its correlates, its footprint in the brain, rather than focusing on things that happened before, or such as attention, or afterwards, such as a report or task setup. Okay, next slide. Now you have to distinguish these NCC from enabling factors, so-called background or enabling factors. And particularly if you're working with patients, that's very often very difficult to do. So these background factors are things like your heart has to beat. If your heart doesn't beat, you will faint within 20 seconds, right? If somebody chokes you, restricts the blood flow to your brain, you'll faint very quickly. Okay, now that doesn't mean that consciousness is in the heart, like the ancient used to think, or that it's in the bloodstream. It's just one of many things that has to operate in order for the conditions that are necessary for consciousness to exist to be there. So in the brainstem, we know, particularly from the work from this gentleman, von Economo, that there's a range of stimuli that have to be present. So they're sort of collectively known as the brainstem arousing system, noradrenaline, acetylcholine, etc., that have to be present, orexin, that have to be present in order for you to be conscious. That does not mean that the brainstem mediates consciousness. We know this because in patients that have an intact brainstem, but that have massive cortical damage due to massive strokes or bleeding or viral infection, they're not conscious anymore. Conversely, tiny lesion in the brainstem can completely knock out consciousness, in particular if they're bilateral. So we know that many mechanisms in the forebrain, in the general arousing system, in some thalamic nuclei, need to be present for you to enable consciousness, but they themselves do not provide the phenomenal content that makes an experience a visual one or an auditory one or painful one. For that, by and large, we need to look, well, what are the structure we need to focus on if it's not the brainstem? Next slide. We have, for instance, 200 million neurons that are in the, roughly 2 feet long spinal cord at the back. And if you lose those, or if you have a cut there, you're in bad shape. You'll be paralyzed, like Christopher Reeve, for instance, here, the Superman actor, but yet you're fully conscious. So that's important conceptually because it tells us it's not just any neural activity per se. It's not just the fact that there are neurons firing, as some people used to believe, that is good enough for conscious. You can lose the entire spinal cord. You might be paralyzed, you're quadriplegic, etc. You have to be, somebody has, a machine has to breathe for you, but your consciousness will not be impaired. Next slide. There's a structure at the back of the brain, a little brain, so-called the cerebellum. It contains, in fact, 80% of all your neurons. Those are the granule cells. Right, of the 86 billion neurons in the human brain, roughly 60 billion or some 65 billion of them will be in the cerebellum. Some people never had them, so this is a rare case of a cerebellar agenesis. It's a Chinese woman. And she had some infection. She had to go to the hospital and she got scanned, and then people realized that she was born without a cerebellum. More common is you might lose your cerebellum due to a stroke or a tumor, in which case you become, you may become ataxic, you may walk, you may be very difficult to speak, you may look like you're drunk, etc. But these patients do not complain of loss of consciousness. Patients with cerebellar lesions, for instance, following neurosurgery, do not complain that they can't see anymore, that they can't hear anymore, that they can't feel pain or pleasure. In fact, if you talk to them, they're very difficult to notice any difference in their conscious experience of the world. Of course, they're in a different state. There are many things they can't do anymore, but their conscious experience, their mind does not seem to be directly affected. So again, that gives us a very important clue. These are very complicated neurons. The neurons in the cerebellum, particularly the Purkinje cells, among the most, they're these paradigmatic, beautiful neurons that have extended, they look like coral. They have this beautiful action potential that goes all into their, and calcium spikes into their dendrites. They have memory, they have representation, they have everything one might think a brain needs, but you remove the cerebellum and consciousness isn't affected. Why might that be? Next slide. So I think that gives us very important clues. Click again. So as far as we can tell, at least in us and in closely associated creatures, that doesn't mean in all creatures, but in humans and in other mammals, it's the neocortex that's most closely linked to consciousness. So neocortex is really it's really a tissue. It's like a pizza. You can conceptually unfold it, certainly using sort of computational techniques. You can unfold it. It's roughly the size of a pizza, 14-inch cortex. And it's as thick as a pizza with toppings, like 2 to 3 millimeter. But of course, it's highly convolved, highly convoluted, and you've got two of them, one on the left and one on the right. So this is on the right. You can see the image of a macaque monkey. It's very similar in us or in other creatures. And we know that if you lose cortex, or if you lose the usage of cortex, then consciousness has left. We also know from 150 years of functional neurology that losing particular aspects of cortex can give rise to loss of specific aspects of conscious content. And furthermore, if you stimulate cortex in specific region, you can induce specific percepts, like a specific visual percepts, auditory percepts, ecstatic experiences, etc. Next slide. So it's really cortex that seems to be the stuff that gives rise to consciousness. The evidence, as I just very briefly summarize, this is typically an entire series of lectures I give at university, is based on lesion studies, i.e. if you have a stroke or gunshot or virus or what other reason that causes you to lose particular parts of cortex, you can lose certain attributes of consciousness. Stimulation studies, very often during neurosurgery. There was just a beautiful study out of Stanford that came out. Or using external techniques, non-invasive techniques like transcranial stimulation, or more recently using much more finely refined optogenetic stimulation. And of course, but the evidence is weaker there because it's not causal, recordings and neuroimaging studies. So recordings, EEG recording, MEG recording, ECOG recording, electrode recordings, fMRI, PET, all the gamut of modern tools that experimentalists use. Although the caveat there's always those are correlation versus the first two categories are causal studies, lesion and stimulation. Summarizing all of that, next slide. So here, for instance, I show one experiment comes out of Stanford, the lab of Joseph Parvizi, where so here you're seeing the brain from below. So it's the undersurface of the brain, you know, just above the cerebellum. The front of the brain is on top. The back is on the bottom. And here people had to put in electrodes to monitor these patients for possible surgical resection due to epileptic seizures. So you can record from those electrodes, and you can see that those electrodes are selective for phases. These electrodes are just underneath the fusiform phase area that we know from now 15 years of fMRI, again, respond when you do phases versus non-phases, phases versus scrambled phases. And so that's not surprising. What they could do here, because these are electrodes on top of the surface of the cortex, you can also stimulate them. And then what the study reported that when patients is looking at a phase, for example, the phase of the experimentalist or the doctor, and you stimulate them, you can get distortions of their facial percept. So you seem to interfere with certain aspect of facial processing. It's very specific. It's on the right side in the vast majority of patients. It's on the right side. So the left side, even though you still have the correlate, the fMRI or the EEG still responds to phases. If you perturb the left side, it doesn't seem to directly affect the conscious percept of a phase. So this tells you that the conscious experience of a phase seems to be closely associated with this area. Now that's of course still very crude because we're talking about 1 billion neurons. And now you really want to find out which of the 100 different cell types in this particular region, or which specific circuit in that particular region is necessary for consciousness. Next slide. Yeah, so there's a vast, we wrote a couple of review articles about it, which I just want to summarize. So in view of this data, particularly the clinical data from lesion studies and from stimulation studies, primarily from patients, from humans, seems to indicate that conscious experience is associated with what we call a posterior hot zone. And at this point, not very precisely circumscribed region of cortex that involves aspect of the temporal lobe, the parietal lobe, and of the occipital lobe. That activity in this part of cerebral cortex seems to go hand in hand and seems to be causally necessary for conscious experiences. Now, the note of caution here, of course, that the vast majority of study involve vision, somatosensory, and audition. There are some studies involving willful initiation, voluntary action, and they also involve structures in the parietal lobe. Of course, the universe of conscious experiences is much broader, and so one has to do this for each and every modality. Of course, what is not shown here is activity in the front of the brain that traditionally is associated with higher order thought and reasoning, moral reasoning, intelligence. And of course, there is evidence in favor, particular functional evidence in favor of the involvement of these prefrontal structure. But to my reading of the literature, that is involved in pre-processing or post-processing, like planning, task, memory, and task execution, rather than directly in the causal, in actually consciousness for those particular attributes of the experimental setup. This also points at a possible dissociation between consciousness on the one hand and intelligent or higher order actions on the other, that they may not be the same. They may co-vary, but that intelligence is really something quite different from conscious experience of the world. Next slide. So this gets us to the problem of other creatures. So in general, of course, there's a problem of the other. How do I know any of you are conscious? I really don't know. I have to abduce it, right? So abduce it is a form of inference. Given everything I know about the world and about the world as taught to me by science, I infer that all of you are conscious, because otherwise I would have to presuppose one set of law, of psychophysical law for my brain and a radical different psychophysical law for the brain of everybody else. That's extremely unlikely. For the same reason, I can abduce consciousness here in others. In this case, the other is my daughter and her German Shepherd. And so we can abduce consciousness in other creatures. This inference is easier the more similar structurally those other subjects or objects are to me. If the brain is very similar, then that's easier to make than if the brain is much more different. So that's why, as biologists, most of us assume that mammals, by and large, are conscious. Why? Next slide. Similarity of behavior, similarity of brain architecture, evolutionary. We know we are all closely related. And the point I want to make here, so at my institute, we're studying. Maybe you can go forward. Yeah, so here you can see on the lower left, we study two creatures. We study the brains of Homo sapiens and the brains of mice. They differ by pretty much exactly a factor 1,000. So in terms of weight, in terms of volume, in terms of surface area, and in terms of numbers of neurons, it's roughly a factor 1,000 difference between the mouse brain, the tiny brain there, and the human brain. Yet if you look at the individual components of the brain, neurons, nerve cells, in this case, pyramidal cells, but we've done it for lots of other cells. We have a big Nature paper coming out where we describe this in great detail using transcriptional and morphological technique and electrophysiological techniques. If we compare human and mouse brain and we account for the size differences by here simply removing the scale bar, they look remarkably similar. They're not identical, of course, but they're remarkably similar. And so certainly for mammals, the biggest difference among mammals is, this factor 1,000, or of course, we don't even have the biggest brain, right? There are elephants and whales that have even bigger brains. So over all mammal species, there's this factor of 50,000, 60,000 that differs, but that's sort of quantitative. And certainly their behavior, they don't talk, of course, but human babies don't talk and people with strokes don't talk. The main thing that we have is language, true language, and a hyper-developed sense of our own importance to the world. That's really what makes us different from most other creatures. But certainly, if you look at it from a point of view of the physics of the brain or the biology of the brain, it's remarkably similar, and so therefore we assume that certainly mammals are also conscious. Now, this inference, next slide, is more difficult to make as we go farther away from the mammalian brain. So this leaves all of these questions. There's a bunch of really good, interesting questions we can pose, like why is it if it's experimentally true that it's neocortex that's primarily involved in consciousness, why neocortex? In particular, if it's true it's the posterior heart zone, where's the difference between the back of the brain and the front of the brain? There are massive differences in the connectivity. What precise aspect of connectivity matters here for consciousness? Why not the cerebellum? If you believe that neural activity, particularly synchronized or oscillatory neural activity, is irrelevant for consciousness, why not during seizures? Typically when people lose consciousness during seizures. Next. Why not cortex during sleep? Why not cortex during sleep? Neurons are still active. It's not that they sort of shut down. They don't dissolve into soup. There's still connectivity. Why is cortex not generating consciousness during deep sleep? How does, what about near-death experience? How do near-death experience come about? In a case like the one on the lower right by Nico Schiff, here we have islands. This is the PET images. Here we have islands of cortex that are still active. Is this person experiencing something? Difficult to answer question at this point. Next slide. Maybe you can just click through. When does consciousness first start? it's an experimentally very interesting question. Are you first conscious when you're a fetus, or are you first conscious when you emerge into the world and you have to start breathing on your own, when you're actually born? What about sleepwalking? Is that a conscious state? What about, there's some very strange subjects around. This is an instance of Craniophagus twins that live here in Canada that are connected at the level of the thalami. So they have connected, essentially have one contiguous nervous system. Are they one or are they two subjects? Is consciousness true for all mammals? And then what about animals that are very different from us, like octopus? I just saw this beautiful movie, My Teacher Octopus, or here bee, very complex creatures. They have 1 million neurons compared to 86 billion, but they have very complex behavior. They have this simple waggle dance. They can communicate to their sisters, the source and the location of food. Their circuit density is 10 times higher than the circuit density of a human brain. Does it feel like something to be a bee? Next slide. And still more question, what about cerebral organoids? We can now build, using advanced tissue engineering technologies, we can build these so-called mini brains in a dish, brain organoids that are 1 million neurons that have maybe your DNA, that have neural activity. Does it feel like something to a cerebral organoid? And if not today, what about 10 years from now when we can maybe grow them in carpets and we can co-culture them with other neurons like thalamic neurons? At what point does this tissue assembly feel like something? What about a group mind? And then of course the question of the ages, what about a robot? This is from Ex Machina. Or what about Alexa? Alexa is very complex. Alexa can speak. She might have what Aristotle would consider rational soul. Is Alexa conscious? You can ask her, she'll tell you. Next slide. So for that, you need theories. And so now let me come to the question of theories. There are many theories, of course, around, or there are many things that people call theories. By theories, I really mean a well-developed, starting with some concept, a very well-developed body of knowledge that's organized in a coherent way that gives rise to very, very specific prediction. I don't mean when people say, oh, my theory of consciousness is it must be layer 5 neurons, or my theory of consciousness it's 40 hertz. That's not a theory, that's a working hypothesis. That may be perfectly fine, it may be a correct working hypothesis, but that's that's one working hypothesis, that 40 hertz is one of the hallmarks of consciousness, which turns out not to be correct, but that's not a theory. So in terms of well-developed scientific theories, they're really, of consciousness, there are really two. One is the global neuronal workspace theory of Stan Dehaene and Jean-Pierre Changeux, that traces its origin back to Bernie Bars, the American psychologist Bernie Bars. Its key assumption is that consciousness, it really traces its way back to the fact that whatever I'm conscious of, when I'm conscious of something, pain or pleasure or seeing, that information is accessible to all my systems, in all my cognitive systems. I can talk about it. I can plan with that information. I can use that information to retrieve memory. And so here the GNW postulates that consciousness is a type of information in the brain involving the selection of information for this global broadcast. So when information is just used locally, let's say to control my eyes or to control my hand, grasping my coffee cup, then that information, it remains encapsulated. If it's not broadcast, it doesn't become conscious. Only information that's broadcast to all the rest of the brain, that's what, by definition, that's what consciousness is, when it's broadcast to everyone else. Okay, next slide. They have particular systems in the brain that are necessary. So here there was a recent experiment in Science where they showed some evidence in favor of this. The idea is when we have information, so this is a psychophysical threshold experiment where you show something and sometimes you see it, sometimes you don't, just like I showed you earlier on. And here the idea is when the information can propagate, but if it's weak information, if it's not, let's say, because you're not attending to it, it doesn't make it into the structure in the frontal of the brain, into what's called the global neuronal workspace. But once information is, because let's say you're tending to the information on this particular trial, the stimulus now gives rise to a more vigorous feed-forward wave that propagates through, in this case, the eyes, the LGN, primary visual cortex, goes through higher order cortex, goes into the neurons of the workspace in the prefront, and is then broadcast back to all other relevant structures of the brain, in particular the parietal, occipital, and temporal structures. And in that broadcasting, you become conscious. So that's global neuronal workspace. Next slide. The opposite, a different view, a fundamentally different creature is of a theory, is integrated information theory that Paavo mentioned already and by Giulio Tononi. It's postulated, it's sort of a much more axiomatic theory. It proceeds to say, well, what are the properties, sort of the transcendental property that any conscious experience has to obey? It exists for itself, it's definite, it's specific, it's one and it's only one. And from those 5 axiomatic properties, it deduces, postulate that any physical system, any mechanism in a state, whether that's neurons or transistors or the sun or anything else, has to obey in order to realize these 5 transcendental properties of consciousness. And ultimately, it associates consciousness with intrinsic causal power. That's really what it is. You have a system, any physical system, if it has what the theory called maximal irreducible intrinsic causal power, which you can quantify by a number called phi. So phi specifies not the entire, so the theory itself says that this intrinsic causal power unfolds, you can unfold it in this gigantic conceptual qualia space. And that ultimately it's a physicalist theory. It says that there's this complete identity, one-to-one mapping between this intrinsic causal power that's unfolded and conscious experience. So any aspect of your conscious experience, that it feels painful, what exactly does it mean to feel painful? Or when I'm looking at an empty whiteboard, that even an totally empty board feels like something, it feels spatially extended. That feeling has to map completely one-on-one onto the underlying high-dimensional unfolded structure in this qualia space. But it quantifies that using phi, this number phi. It's a number. It's not measured in bits because it's not information. So here the information in integrated information is this older usage that Paavo alluded to, informare. Sort of, it's not Shannon information. It's not bits of information. It's to give form to something. And so according to IIT, ultimately, the fundamental constituency, no matter what they are, ultimately they're causal powers. So it's in some sense, and these causal powers inform the system, and that's what it feels like to be that system. So it's sort of, you can think of it as an Aristotelian form of hylomorphism. All right, so you have a system, it's in a particular state, these neurons are on and those neurons are off, or you have a bunch of gates, transistor gates, and these transistors are on and these are off. And then this state, if it has some intrinsic causal power, if its phi is different from 0, will feel like something. For any system, it's quite radical. It doesn't have a threshold. Any system that has a phi different from 0 exists for itself. Therefore, it is conscious. It is irreducible. So phi different from 0 means it cannot be reduced to its simpler component. How much it is irreducible, that's quantified by the amplitude, by the magnitude of phi. So if you have a very high phi, if your brain, for instance, in that particular state has a very high phi, its maximal irreducible is very large, and therefore it's very conscious and has lots of different distinctions. If you have a very simple system, let's say a paramecium, which still consists out of 10,000 different proteins, of 1,000 different types of proteins, still of immense complexity, but it may not even have a brain. It's just a single cell. It still feels like something because it has some irreducible causal power. All right. Now, how do you test these theories? These are very different theories. And how do you test them today, not in some future time? Well, so next slide. Oh yeah, so the IIT has come up over the last 20 years, a very exciting development that's now being tested in a variety of clinics in Europe and here in the US, because it says any system has to be highly differentiated and integrated, in particular to be conscious. In particular, when I look at the cortex, I can look at the activity of the cortex on EEG, and I can measure how differentiated and integrated it is by perturbing it. Okay, so experimentally, what do I do? So I have people like these patients where I simply don't know whether they're conscious or not. There are thousands of patients like that on the world. They're sort of in a vegetative state or in a behavioral unresponsive state, and I simply don't know because they don't respond to spoken command. When I say, can you please open your eyes or move your limb or flutter with your eyes or anything like that, they don't respond. They're alive. That's not the issue, but are they conscious? Or for example, during anesthesia or during catatonia. So on all these cases, what you do, you put a high density EEG on their brain and you sort of knock their brain by using transcranial magnetic stimulation. You zap their brain, essentially TMS. And then you can perturb the brain, you can measure the perturbation, and you can derive the complexity of that using Lempel-Ziff. Measure. And so essentially you're asking how, if I look at the brain EEG response on this high density EEG, let's say 128 or 256 channel, how compressible is that EEG? It's a final pattern of activity. If it's very compressible, the system is much simpler, and that typically tends to be associated with deep sleep or with states of deep anesthesia, surgically induced, or with coma. Or is it less compressible? So they compute a single number called PCI, Perturbational Complexity Index, and empirically it's been measured now in several hundred patients. Whenever the PCI is below a threshold, the patient does not seem to be conscious, and when the Perturbational Complexity Index is above a threshold, the patient, the subject, whatever, is conscious. So that's really a tool that's now been developed to measure the presence or absence, to detect the presence or absence of consciousness. And that shows you that there is progress that can be made on these difficult mind-body problems. Next slide. Yeah, here, so here I'm measuring myself. I'm being measured. You can see my brain response in response to the transmagnetic stimulation pulses, so I can tell you I'm conscious, at least according to the theory. Next slide. Yeah, and here this is done in patient. This is done, as I said, for some of those patients where this can be used now with 100% specificity and sensitivity. So it's really exciting to clinicians. Next slide. But so there's this ongoing experiment, a so-called adversarial collaboration, to test contradictory predictions. So it's a really interesting process. So we got together at my institute, the Aalto Institute for Brain Science, several years ago in Seattle. We being a bunch of psychologists, experimentalists. So there was Giulio Tononi and Stan DeHaan, David Chalmers, and Francis Fallon, philosophers. It's led by the PIs, Lucia Meloni. She's in Frankfurt, Liat Mudrik, she's in Jerusalem, and Michael Pitts, he's here in Oregon. And we got together and then over the next year elaborated a detailed experimental paradigm, 2 experiments with 500 subjects each using MEG and EEG coupled, using fMRI, and then using in ECOG in patients, epileptic patients, ECOG arrays. Well, we're doing 2 experiments. All the data will be uploaded. Each experiment is done by 2 separate labs. Neither of the labs is associated with IIT or GNW, so it's done by independent labs. Half of the data will be blinded and locked away. Half of the data will be used to test, localizers and predictors, and then they'll be tested on the data that will then be unmasked. And then all the data will be made accessible to everyone. So it's really a pretty cool thing. It's funded by the Templeton World Charity Foundation. Next slide. Well, yeah, so it's really an experiment in the sociology of science also, because it's a large experiment involving 12 labs, 50 people all across the planet. So there are two experiments. This is the easier, the simpler one. So this really is a variant of Haruki and Manley. It tests, I don't really have the time to go in detail. So it essentially tests so the challenge in this adversarial collaboration, because we've submitted the protocol already to, it's already submitted now, and we can't change it anymore, is to come up with an experimental design where the outcome, we can talk about the different outcomes, where people agree, if outcome is A, then this will support, let's see, IIT. If the outcome is B, it'll support G and W. And we did that for all the different aspects. And we essentially, it's the prediction is essentially around where is the neural correlate of consciousness in the brain. Is it essentially the back of the brain, this posterior hot zone, or is it in more frontal structures? When does it arrive? And how long does it last with the duration of the stimulus? So you see, if you have a stimulus that you're conscious of, the stimulus is clearly present. So here you always have stimuli that are individually always present, but sometimes they are, and there's only, in this first experiment, there's only one stimulus ever present on the screen. It's either present there for 250 or for 500 or for 1200 milliseconds. IIT says, as long as you're conscious of it, there will be a neural correlate of consciousness in the posterior hot zone. While G and W says, no, it's only when you broadcast it first that there is this broadcasting wave of activity. If nothing changes, there's no need to update anything. So there's a number of clear predictions where the theories differs in terms of where's the footprint of consciousness, when does it happen, and how long does it last. All right, so those are ongoing experiments. They'll take the next 3 years, and who knows with COVID how long that'll be delayed. Next slide. Yeah, so I just wanted to come to my end. The theories makes two radical different predictions, which is really interesting. So global neuronal, when it comes to artificial consciousness, global neuronal workspace argues that consciousness really results from particular type of information processing computation. As long as they're physically realized, they need feedback, you need this global operating architecture, this global broadcast architecture. When that is realized in appropriate hardware and software, the system will be conscious. So, you know, I paraphrased and say experience is just a clever hack away. You build the right hardware today using today's technology, you'll get conscious systems. IT argues that ultimately it's about causal power, and that if you look at this, the computers, von Neumann computers, right, with the conventional gate architecture and the very low connectivity that you have in a typical CPU, with very little overlap between the input and the output, radical unlike neurons. Computers essentially, although they can of course and will soon be capable of intelligent behavior, will not feel anything at all, because they don't have, unless the system is endowed with the same intrinsic causal power as the human brain, it will not be conscious. And digital computers of the sort that we build today have a tiny amount of intrinsic causal power because of their connectivity. So you can build artificial conscious machines using sort of neuromorphic architecture, using essentially the causal structure of the brain. But digital computers, even though, next slide, they may be as complex as the human brain. So it makes this prediction that even if you build a computer simulation of a human brain, if it's, you know, on the cloud, even if it wakes up and says it's conscious, it's all fake, it's all make-believe. Just like you can, you know, simulate, you can solve Einstein fields equation for the black hole at the center of our galaxy, you know, Sagittarius A star. The Nobel Prize was just handed out for its observation. Yet you don't have to be afraid that you're going to be sucked into the black hole simulation, right? Because it doesn't have the causal power of gravity to bend space-time around it. It simulates the causal effect of gravity. It simulates a black hole, but it doesn't have the causal power of black hole. Same thing, if you build a human brain simulation in 10 or 50 or 100 years, that simulates accurately a human brain, that doesn't mean you have created the intrinsic causal power of a human brain. And so it means this system will not be conscious, although it looks and it speaks like it'll be conscious. So it's all a deep fake. And I think that's my, what's the next slide? Yeah, I just said that. Next slide. Yeah, and so therefore, I just wanted to end with this view. According to IIT, one really has to strictly separate intelligence and consciousness. So in creatures that were evolved like us, you can show in computer simulation, there's this nice relationship between the adaptability of the creature and its integrated information. The more the system architecture integrates information from its different sensors with its memory and its planning, the higher the phi. So there is this covariation in evolved creatures. The more intelligent they are, they're more conscious they become. However, you can click. Yeah, you can engineer creatures that are completely dissociated. So as I mentioned, you can, for instance, engineer cerebral organoids. In principle, you can engineer very large mats of them, maybe not now, but in 10 or 15 years, the technology is moving very rapidly. So in principle, they might be quite conscious, but unless you build sophisticated output behaviors or output structures, there's no behavior, right? So by that measure, they're not intelligent. Conversely, we can simulate, and we are simulating more and more powerful software systems, like AlphaGo, for instance, Deep Blue, AlphaGo, etc., that have very high level of intelligence, potentially at least, yet they don't feel like anything. And so when we really think, we as biological creatures, we lie on that diagonal where intelligence and consciousness co-vary, but that doesn't have to be true for our creatures that we build ourselves. And with that, I thank you for your attention. Yeah, and I've written about all of this in my last book, The Feeling of Life Itself.
Dr. Koch, thank you very much for your very inspiring lecture. We look forward to seeing the results of your latest and very brave projects. You made a very convincing case of consciousness being correlated with cortical activity, but even if we're inclined to believe it, we cannot take it as this correlative evidence as a cause to defend a causal relationship between neuronal activity and consciousness. These neuronal networks may just be structures that are necessary for others to infer consciousness from us, but the actual basis of consciousness may lay somewhere else, we don't know. Do you have any proposals to address this fundamental problem on the basis of consciousness besides the projects that you're already involved in? Why do you think that a conscious AI with an in silico basis would only be a simulation of the human consciousness, even if that continues to evolve without humans potentially? Would a conscious brain organized convince you?
Well, so those are, okay, so you asked many different questions. So experimentally, even if, so let's say it's true, it's cortex, let's say even it's true, it's posterior hot zone, then of course, that's just the next step. We have to find out, there are 15 billion neurons there, which are the essential neurons. And of course, now with modern molecular biology, we can turn on or off individual connections. So we can really find out, particularly in experimental animal, is it the feedback? What type of feedback is involved? What are the individual actors? So we can drive this experimental program to an unprecedented level of resolution. Brain organoids and computers, it's much more difficult to answer the question unless I have a theory. If I just depend on my intuition, well, sort of it doesn't feel conscious. Right, that's not good enough. So therefore, I first need a theory like GNW or global or IT or Bohm theory of active information or whatever. I need a theory that I first need to validate in the only subjects where we're really sure we're conscious, which is humans, adult humans that can tell us about it. Myself, of course, first and foremost, but then you and other people that can directly tell me about it. So whatever theory I have, I need to validate it in humans. then in closely related species like monkeys and cats and dogs. And then as I become more confident in the prediction of in the power of the theory and the ability to infer consciousness in others, I then can take it, well, what about an organoid? Or what about a computer? But you can't do it the other way around. So you have to start with a theory that's developed and validated around the only subject that we know are conscious for sure, which is us.
Yes, thank you very much, Dr. Koch. And we will now have a short 5-minute break, and we will get back soon with a panel discussion on consciousness.